So Peter asked me about a week ago to do this talk for him. He had to be out of town for today and tomorrow, but he'll be back. So if you have a lot of questions, when he gets back, you can ask him. Got it. So let's kick this thing off. A little bit of history where the stereotactic approach came from. One of the earliest reported that Peter and I could find was from Horsley and Clark in 1908. This was in rhesus with a millimeter glass plate, and they used this to guide surgery of cerebellum, and we thought that this would be a very nice introduction to show you that stereotactic approach has been around for a long time. Now let's jump forward a bit. A couple of atlases that have guided our field quite a bit. The 1967 Tayloric Atlas had several brains and led us to an approach for a method for aligning brains for averaging. And we see in, whoops, I hit the wrong button. Go back. Here we see some averaging looking at SMA functional response distribution across multiple brains after normalization. And over here we see some sulcal probability maps. These are sort of some early things, and these are like two-dimensional projections, not what we're used to working with today with full 3D images. But we've based what we do in the future now on these approaches. One of the earliest ways to spatially normalize brains was developed at WashU by Peter Fox, Joe Perlmutter, and Marcus Rakel, and they used a sort of a, I keep hitting the wrong thing. They used a little grid and they x-rayed it and they came up with a schema to find the ACPC line from the skull landmarks. Of course, we have a lot better ways of doing this now, but this was an early stage. And Peter was telling me that while this worked quite well, people didn't get very excited about it because they didn't see any real advantage in it until they published this next paper showing that in a spatially normalized space, you could improve the signal-to-noise ratio or reduce the noise and detect things that, just like you did in image averaging with repeated images, but in this case across images using spatial normalization for alignment first. And we see, whoops. I'm getting it. Back one. So we see how much the noise level drops off. There's a predicted level and then a measured level, but we know when we average across these images, and these were PET images, so they're relatively noisy. So what does it mean to adjust a brain to the Tayloric Atlas brain space? Here I'm showing you the general schema. We have to do something to orient the brain to match the brain in the atlas, and then position and size. And this approach has been around for many years, and early on it was done manually, and where people would identify the mid-sagittal and the ACPC manually and then adjust the brain to a fixed size. Now we use automated approaches. But the motivation here was a little beyond what Peter was showing you, that we could just average and get better signal-noise ratio and report findings. There was a lot of information that you had access to in this particular brain atlas, in particular Brodmann area labels, in which you could address by coordinate. And we'll see later on how we do that nowadays. So this is one of the first really big features that added new processing capabilities for our neuroimaging approaches. It incorporated spatial normalization, but we call it statistical parametric mapping. It was introduced by one of our keynote speakers, Carl Friston, and in the group. And so I want to talk about coordinates a bit. And this is sort of when we developed, Peter and I put a lot of time and effort into the BrainMap database, and early on, the best way that we could record findings was by coordinates in a standardized space. And I'm showing you these coordinates because not everybody got it right. If you'll notice, in the right M1, it says x is negative, when in fact, this is a right-handed coordinate system, plus x, plus y, plus z. And so whenever we put coordinates into the database, we actually still have to filter this once in a while. They get left and right backwards. And why do you think that is? There's this neurological and radiological approach to looking at images where the left and right are on opposite sides. That's what we think anyhow. So in terms of using coordinates, we got a little more advanced, and here's a case in which they're showing not just the location, a number of clusters here with different kinds of tasks, and they're showing you a coordinate that associates with it. When it's a cluster, it's usually just the maximal z-score. But also, they give us the size, and because we knew the location, we could look up a Brodmann area, the BAs, and then a description. So coordinates sort of gave us a pointer into other things, but they could be combined with regional results as well, which were reporting the centroids of regional changes. And we kept advancing and advancing, and if you take a look at coordinates, this is a large number of coordinates just taken from the BrainMap database, plotted on standard brain that we use for Taylor-X space. It's called the Collin brain. And you see that most of the time, they actually fall within the brain. And most of them are in cortical regions, not necessarily all of them. So this was another major change in the way we could approach, another tool that allowed us to use coordinates in a very interesting way. It was introduced in 2002 by Peter Turkeltaub, and he called this method activation likelihood estimation. And what he did, he looked at a number of PET studies based on speech, and at each location, instead of using the coordinate, he generated a 3D distribution with a Gaussian function for each coordinate. So it spread it out into a more continuous distribution that he was able to threshold and produce these activation likelihood estimation mappings. Now, this was interesting, but Peter told me, I thought when I first looked at this, he was validating it with fMRI. And what Peter told me was he was validating the fMRI with the BrainMap data, that this is one of the first cases of showing that PET results and fMRI results were somewhat consistent. Oops. So the ALE method is extremely popular in our field, and we see there's been a tremendous number of people who've added to it, and you might notice at least one other person sitting at the desk over there in this list, Simon. But Peter started off in 2002, Angie Laird in 2005, and all the way up 2016, and there'll be more discussions on this kind of approach. So this methodology has been refined over the years. And look what can happen if you come up with something really, really novel, and it was novel. In 2017, Peter Turkeltaub won the Norman Jeskin Prize for Behavioral Neurology. 15 years later, so don't get in a hurry right after your major discovery. So we have him to thank for kicking this off, but a lot of people have benefited from this approach. So let me talk a little bit more about the BrainMap database. Peter and I have worked on this for many years. We started back in the late 80s, so we've been working with it for 30 years or so, and we only had coordinates way back then. But why could it be a good environment for meta-analysis? Well, we start off, we have a bunch of coordinates, and the database organizes things that the coordinate data literature. So if they don't report coordinates, it doesn't go in the BrainMap database. And just to give you an update, we've put now over 3,000 papers completely coded, more than 16,000 experiments documented with over 75,000 subjects and over nearly 130,000 coordinate locations. There's a lot of stuff in this database now. And so you can actually use it to go back and search it for various things. More recently, we started adding in structural data, which is mostly about disorders or lesions, and you can see the numbers we have here. So you can actually go to the BrainMap website and become a BrainMap person. You can download this software called Scribe. It is the software that allows you to enter experiments, and it's free to download. And when people enter these experiments, when they get finished, it gets submitted, and it goes through a quality control review at our site just to make sure. And sometimes we have to get back and forth several times. We also have a search engine, which we call Sleuth, which allows you to find and download BrainMap data for subsequent analysis. You get to pick what you want to download and set up search criteria. And also we have this neat named ale called ginger ale that will do ale for you based upon searches. So let's move along here. Maybe. Oops, where did we go? This is just a description of the BrainMap database structure a little bit. I want to move to this one showing just under the behavioral domain all the kinds of things that got coded in here. And then here's a search that you could do. This search was done with cognition, language, orthography, and it created an ale and overlaid it onto an anatomical image. So you can do this using the BrainMap database structure. You can also, so this is a search based on behavioral domains. You can also do searches based upon paradigm class or experimental designs. This is another type of approach using the BrainMap database. Janie Robinson did this. She was interested in connectivity, functional connectivity of the amygdala, so she put a 3D region of interest into TaylorX space and searched for coordinates falling within that region of interest that also in the same experiment had a coordinate elsewhere. And so she reported all of these co-coordinate things as analytic connectivity. So we can see what happened for left amygdala and right amygdala, and showing you that the cytoarchitectonic parcellation of amygdala looks a lot like the parcellation that you can actually produce through searches in the BrainMap database. Quite interesting that we can go backwards from all these coordinates. Another paper that was really important showing that the similarity between resting state fMRI and BrainMap data using ICA decomposition. Most of us know how to do ICA decomposition with resting state fMRI or any fMRI, but with the BrainMap database, we had to create each frame of time was an experiment with coordinates. And so they did a very long ICA, but they limited it to 20 components. And if you look at this, there's a pretty good match in the visual. There's the default mode network. So the resting state network's on the left, BrainMap's on the right, all the way through these networks. And then they went back into the database and looked at were these networks, what kind of behaviors were associated mostly with them. And so if you look at the visual ones, we see a lot of perception, vision, motion, shape, and things like that. And if you look at the one that's action execution, that happened to be somatosensory cortex, and so forth. We've updated the approach. This took them several weeks to go through the database to do this. Now you can do it online with software called Mango. And here's the default mode network and the behavioral analysis results you get online. And so looking at the default mode network, we see up here social cognition was the highest ranked, and we rank them with z-scores. And if you look at paradigm analysis, this is the experiment that is most likely would activate or lead to coordinates in those areas. So Peter thinks that paradigm analysis might be the most important thing here, that people might be wanting to know how to activate a certain region, and so you can put that region into Taylor-Rack space and then do the paradigm analysis to tell you what kind of experimental designs to use to activate it. Another graph theory comparison showing something coactivation by meta-analysis, coactivation by resting state. Again, we're just giving you comparisons. And here's one that was done with Angie Laird. She used ALE to get nodes and then Macum to get edges and a little network, and then broke it down into various activities: emotion, action, and perception. And this is another version of that one. And quickly, I want to get to the voxel-based morphometry side of things. So this is how we code those kinds of papers. And this is a result that came out. This is the second most highly cited brain article in 2014, indicating that hubs, MRI lesions preferentially attack hubs. We won't go into the details here. It's a little confusing to try to present all of that, but it was a quite popular paper. Here's another one, identification of common neurobiologic substrates for mental illness. This one was kind of interesting, looking at all patients. This is kind of a meta-analysis, and then looking at psychotic, non-psychotic only, and they found common areas. And then they looked over here at where the controls were greater than patient. And this is a long analysis. But again, we're looking at coordinate-based analysis. So this was a very sort of adventurous one to compare the ICAs we could get from voxel-based morphometry with those from the functional database. So I showed you there were two parts of the BrainMap database, and you see that there's a fair amount of correlation between some of the anatomical features, and then we get into auditory, visual, ultimately down to sensory motor. But the anatomical and the functional databases are representing something sort of similar. Oops, let me finish. Ow. Well, I think it's a good place to stop anyhow. So I'm sorry that Peter couldn't be here to, there we go. So this is very complicated to look at. Yeah, I know, time's up. This is very complicated to look at. So we created just one more of these. So this one shows diseases. Notice the graph on the top right. These are diseases that associate with this area. It doesn't say that this area of the brain has a disease. It just says that those diseases have been reported with high likelihood in this area. And we'll leave it at that. So thank you. And we're saving questions for the end, is that right?